So for Mr. Khan, okay, we move to the next presenter, Bapak Mujianto. Are you already in room, Bapak?
Yes.
Okay.
Well, thank you and good morning. Assalamualaikum warahmatullahi wabarakatuh. So, before I deal first my presentation, I would like to ask. It's a fear. It's not. It doesn't record, Pak Yoga, because it's feared that word of conference will ask for a proof of presentation. It's okay. It doesn't record this room.
I will ask the PIC of this room to recording the room session.
Oh,
I'm sorry for the our technical.
okay. So, okay, this time for me. Oh, okay. Yes. I will share screen by myself, Pak Yoga, or by committee. Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. I'm going to develop my presentation with the title Refish Predators Structure on the Candidate Site for Lobster Restocking in Pangandaran and Trenggalek, Indonesia. Call use slideshow committee. A slideshow on my screen, or by it's okay, by it's no problem, or it's have to slideshow to white screen. Okay, yes, thank you. This article I wrote with my colleagues, and this one was part of the research in the past when we are still in a marine affair and fisheries. Next. We can see the picture of the screen. Those are lobster artificial reef that we designed and put in Pangandaran, West Java, and Perigi by Trenggalek, East Java. And we did to put them after we analyzed the water environment, like water quality, habitat, reef fish community, and also based on community. And especially reef fish predator structure on the candidate site. Why did we use the reef fish as one of the important parameters before we placement of the artificial, because we can the predator we can reef fish as predators for lobster fish, and then both among the reef fish and lobster, especially feed states, has a closely relationship. Then, well, in general, we know the total area of Indonesian marine more than 1 million kilometers, and about around 0.62% is lobster habitats. And we know that the lobster is one of the fishes that have a high price. Lobster of the price of lobster from Indonesia is high both in the domestic and export market. So the research aim to study analysis the composition, distribution, and diversity of reef fishes on not only reef fish as a predator and non-predators. So, and we know that in the nature, the association between stock lobster seeds and reef fish included of predators and non-predators is closely related to predator-prey activities, and it needed carefully of the artificial habitats because reef fish to present as predators threaten stocks, lobster seed sustainability. Next. The study was undertaken during May and June 2016 at 7 observation stations in Pangandaran, Java, and Trenggalek, West Java. And those collected of data information is before we did placement of lobster after fishes. We used stationary visualization sheets. And we followed by Sherman et al. 2002, and our stationary area was 25 meters times 25 meters. This area is permanent. During the primary survey, data collection after lobster fish placement, and the area used for monitoring and evaluation after 1 year of lobster after placement. And this like quadrat is permanent survey, and then a placement, and then a monitoring site to take abundance of benthic life, reef fish, and other organisms that have a relation with lobster artificial reef. Next. Data analysis, we use a descriptive statistic, and then analysis of distribution based on result, analysis of the fish composition, abundance, and diversity, and also uniformity and dominance of index as one indicator set for restocking. And the distribution analysis is group, and then after that we interpolate in GIS. Because GIS is a straightforward tool for presenting various criteria data with spatial and temporal scale. Next. Reef fish distribution based on the number, species, and attendance. We can see that the composition of reef fish, both predators and non-predators, at the 3 selected stations in Pangandaran waters. Among 5 stations, such as in Batu Karas, Pananjung West, and East Pananjung, we found 17 families with a composition of 3 families of predatory fish group and 14 non-predatory families. The composition value ratio between predator and non-predator fish was found to be 1 versus 6. Presence composition of predator fish comparing the composition of non-predator fish species was low when with the highest value being NISO analysis from Obdastidae family, and the composition of predatory fish compared non-predatory fish species was relatively low, around 1 versus 9, and the highest species present was Scolops affinis from Nemitheridae family. And the next, ecological index data visualization or REVIS, the analysis research index ranks of both predator and non-predators, and the ecological index result or reef is Pangandaran, and Trenggalek resulted the high diversity is Batu Karas around 3.37, and South Damas has 3 and 47 around it in the water. And the uniformity index our location is in category hikes, more than 0.6, and the resulting demand dominance is in the low category in ecological index. From the results index analysis with log base 10, which is less than 0.3. Next. And this one is number of a species distribution of reef group in candidate location of lobster stocking, and the distribution of its reef group based its function resulted major in Pangandaran water having highest percentage of the number of species at 43.43%, and the target 21%, where indicator of fish was 35.36%, and the major fish good average of 56.63%. Based on the conversion value of Bayer fish group indicator and target, found that the percentage of predatory fish in Trenggalek water is 9.94%, and non-predatory fish around 90.80%. So I think was suitable for lobster fishes placement, and also to restocking of lobster, because the non-predator fish more high than predator fish. Next. And our conclusion is the cooperation of predator and non-predator fish species composition Pangandaran West Java around 1 versus 6, higher than Trenggalek water East Java around 1 versus 9, and the range of predator fish individual number Pangandaran among 1 to 9 species. And then the level of pressure is from 1 to 57 individual, and the reef fish index shows a value diversity index in the moderate to excellent category, and the uniformity index species wool station was high and species dominant. Based on the distribution and composition of predatory fish species, the waters of station between West Pananjung and then East Pananjung in Pangandaran, and also in Stasiun Karanggongso and North Karanggongso are recommended location, and those session in the end of 2015 we make to put of lobster artificialis lobster artificial. Next. And we would like to thank with the recent Institute for Fisheries Enhancement, Ministry of Marine Resources, because the budgeting is from it, and then the researchers are grateful to thank help the data collection, identification, and effort who were effort in improving the paper. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you so much to Bapak Mudianto for giving us an informative and interesting research about fish predator on the net of lobster. And now I would like to invite the last presenter, Ibu Regina. Are you already in the room, Ibu?
Yes, I'm in the room. Can you hear my voice? Yes, I'm in the room.
Okay, Ibu, I hear you. So our PIC will prepare your presentation.
Okay.
You can start
Thank
your
you.
presentation, Ibu.
Thank you, moderator, for the time. Good morning, everybody, ladies and gentlemen. In this time, let me present our research about the pattern of digestive enzyme activity and growth of marine fish larvae, mangrove red snapper Lucanus argentimaculatus. Next slide, please. As the introduction, I will say that the snapper fish is one of the marine fish biodiversity. So its availability in the nature should be kept in concern. But on the other hand, this fish also high economical value. And one of the kind of snapper fish is Lucanus argentimaculatus. We call this species as the mangrove red snapper. And in some places, this species of fish to be the catch target for the fishermen, because this species has a high market demand and has a delicious taste and also contain the high nutritional value. So in that condition, it is worried that the high market demand for this fish will have an impact on decreasing their population in nature, as reported in one paper a few years ago. Therefore, the cultivation of this fish in hatchery is important to be done in order to fulfill the market demand as well as maintains their sustainability in nature. And next slide, please. In cultivation scale, one of the important thing is the feed, because the feed to be the source of nutrient. And in cultivation scale also that the feed derived only from be given to the larvae. So the feed should be accepted and used by the fish larvae. The question is, how can we know that the feed we give to the larvae can used by the larvae? In this research, we observe this from two parameters. One is digestive enzyme activity, and the second from the larval growth. From this figure, we can see that digestive enzyme is catalyze the hydrolysis process the feed as a polymer to be their monomer, so that monomer can absorb by the body cell and then use for the growth of larvae. So if we can see there is the growth in the larvae, it means that larvae can use the feed. Next slide, please. Next slide, please. Based on the introduction, the problems for this research is how is the pattern of digestive enzyme activity of a mangrove snapper, and the second is how is the pattern of digestive enzyme activity related to the growth of a mangrove red snapper. So based on the problems, we have a purpose of this research is to understand the pattern of digestive enzyme activity and to determine the relationship between the pattern of digestive enzyme activity and the growth of mangrove red snapper. Previous slide, please. Yes, okay. As the method, this research conducted in Institute for Mariculture Research and Fisheries Extension located in Bali. And as a source of the eggs, we use the natural spawning of domesticated broodstock that rear in the broodstock tank. Before we use the egg, we make a selection of eggs, and we choose only the fertilized eggs that mark with the transparent, floating, and there is a embryonic development inside. Next slide, please. Larva rearing we conduct in a hatchery at Imbrave by using concrete tanks, 6,000 liter in volume, and every tank equipped with the aeration system to supply the dissolved oxygen for the larvae. We use phytoplankton Nanochloropsis oculata during the larva rearing, and we feed the larvae with the zooplankton rotifers, Brachiornis rotundiformis, and Artemia. Beside the zooplankton, we also feed the larvae with the artificial feed, that is a micropellet commercial feed. Next slide, please. As a sample, we use the larvae as a 5, 10, 15, 20, and 25 days old larvae. And we use 80 to 500 larvae for its age for analyze the digestive enzyme and keep the larvae sample in a deep freezer. For the growth analysis, we use 5 larvae for its age and direct measurement. For digestive enzyme, first we make an extraction, and then we analyze protease amylase by spectrophotometry method and lipase by titration. Next slide, please. Two parameters in this research. First is digestive enzyme activity, consisted of protease, amylase, and lipase. And the second is a growth larvae, consisted of total length and body weight. Data that we get, we display as graphic and correlation analysis. Next slide, please. Next slide, please. Okay. In this result, we can see that the digestive enzyme activity, so that the protease activity at the beginning of observation or in the early stage of larvae, in 5 days old larvae, was still very low. But later, this activity seemed to increase with increasing age of the larvae. And from this feature, we can see also that the results show the protease.
Your time only 2 minutes left for your presentation. May you move to conclusion, please?
Okay, next slide, please. Next slide. So as the conclusion, that the digestive enzyme activity of mangrove red snapper increase with the increasing of the larva age, and there is a very close correlation between digestive enzyme activity and the larva growth. That's my presentation. Thank you for your kind attention, and I give the time come back to the moderator. Thank you.
Okay, thank you very much to Ibu Regina for giving us informative and interesting information about growth pattern for the red snapper. And now we are going to the discussion session, and for that we want to record first Bapak Fadli Zainuddin. May you already join on the room, or Mr. Trump?
Yes, in the room.
Okay, Bapak.
Okay.
Join.
Yes.
start your presentation and place the IC on the PowerPoint for Bapak Adli.
Oke, baik. Assalamualaikum warahmatullahi wabarakatuh. Good morning for all. Thanks for moderator. I will to present result of our research with the title Caulerpa resmosa, chlorophyll in indoor cultivation will varying concentration of NNP fertilizer. I am Padli Senudin. My name is Padli Senudin. My team in the research, Profesor Sri Andayani, Profesor Muhammad Mudi, and Firdaus. Thank you. Next. Introduction. One type of macroalgae that is being developed is Caulerpa resmosa. Caulerpa resmosa is a type green algae found in tropical and tropical regions, as also known as sea grapes. Alga cultivation is carried out in open and closed waters. The addition of nutrient needs to be done in closed water, such as in the addition of nutrient NMP. NMP are essential nutrients for algae growth. In aquatic environment, NMP can be limiting factor for primary production. Next. Caulerpa resmosa has abundant pigment, especially chlorophyll, namely chlorophyll A and chlorophyll B. Nitrogen and phosphate contained in the cultivation media directly impact the chlorophyll content. Optimum observation of nitrate and phosphate will be in line with the formation of chlorophyll. The balance of NMP needs to be considered so that it meets the needs that algae can be utilized properly. The availability of nutrient NMP that is not in accordance with the needs of algae can affect the concentration of chlorophyll. Next. Research purpose. Urea and SP36 fertilizer and NMP fertilizer that are generally used in aquaculture activity. So this study aims determining the effect of providing nutrient NMP using urea and SP36 fertilizers with different combination on the chlorophyll content of Caulerpa resmosa. Caulerpa resmosa source. The Caulerpa resmosa used in this study was obtained from Caulerpa cultivator from farmers in Puntondo Hamlet, Mangara Bomban District, Takalar Regency. Next. Experiment design. Seaweed maintenance is carried out the Breakfast Water Aquaculture Fisheries Center Takalar. Using completely randomized design CRD, wide pipe treatment and tree replication. The treatment used was the ratio of urea fertilizer, M fertilizer, and SP36P fertilizer as follow: A 100 and 0%, B 75 25%, C 50%, D 25 the 70%, A 0 the 100%. Caulerpa resmosa was reared in a basin with the addition of an NP fertilizer according to the treatment. Chlorophyll Caulerpa resmosa was monitored every 50 days. Next. Chlorophyll analysis. Chlorophyll analysis washed out by means sewage is extracted, then the results are measured using a spectrometer or wavelength of 663 nanomicron and 645 nanomicron. Chlorophyll content is calculated by the following equation. Please see PPT. Next. Result and discussion. Chlorophyll content per period. Chlorophyll content of all treatment and the beginning of maintenance increase. On the 50 day, the chlorophyll content decrease in treatment DNA. In the final period of chlorella maintenance, all treatment saw a decrease in chlorophyll content. The decrease in chlorophyll was most obvious in treatment DNA. Next, total chlorophyll. The content of chlorophyll B, Cholera rasmosa, which was obtained with higher in treatment B, C, and D compared to chlorophyll A. Chlorophyll A was higher than chlorophyll B in the treatment A. The highest concentration of total chlorophyll was obtained in treatment C. Next. Discussion. The result of study suggests the chlorophyll cholera rasmosa growth was affected by NMP addition. The optimal NMP concentration was obtained in treatment C, namely by editing NMP using urea 50% and SP 36 50%. NMP added to the media treatment C produced the highest chlorophyll compared to other treatments. Too low or too high NMP content in the media has an adverse effect on cholera rasmosa. Next. Conclusion. The addition of nutrient NMP into the rearing medium using urea and SP 36 fertilizer resulted in a good cholera rasmosa chlorophyll content, a ratio of 50% urea and 50% SP36. SP36. Next. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much to Bapak Udayana for giving us an informative and also information about the program and possible ratio for the part. And now we are going to the discussion session, and I would like to have 3 brief questions from the participants who joined us in room by Zoom or who joined us in offline in room here. So may you raise your hand or write your question on the paper or on the chat room for the presentation in Zoom. And also please mention your name and your question and to whom your question is. The discussion session will 10 minutes. Okay, so we have a first question for Bapak Fadli, I think, from the participant in our group from Udayana University student. So the question is why the treatment fee was the lowest chlorophyll. So Bapak Fadli, may you answer the question and tell us about your research work. Bapak Fadli, are you staying in the room? Atau Bapak Fadli? Okay, and we have a second question for Bapak Widianto. And the question is what the predator or banana population in Pangandaran and Bangladesh. So when you tell us about the kind of the predator, please, Bapak Widianto.
Yes, thank you. Thank you for offline of participant. Yeah, thank you for your question. So in Pangandaran, that include of as refish as a predator, fish predator for lobster seeds is Naso annulatus from Blastidae family, and then in Trenggalek, the refish that include as a lobster predator predator for lobster feed is a Scolopus annulatus. Place about the species is for are for lobster seeds, not adult lobster. That's all for me. Thank you.
Okay, thank you, Bapak Widianto, for answer. And we have a third question from Bapak Ngurah dan Dewi Aser to Ibu Puturate. And the question is what the kind of species of seaweed which have the most powerful carbon absorption? Do you use wild type or first generation of seaweed for the research? So, two questions for Ibu Puturate and for Ibu Rate. And you still in room?
Hello. I answer the question now.
Okay, please, Ibu Ratih.
Okay. Thank you, Pak Yasa, for the question. So, what kind of species of seaweed which have the most powerful carbon absorption? For this question, I cannot tell the exact species because in my research I only used one species, which is Euchema cotonii. So I can compare this species with other species. And and for the second question, do you use what type of seaweed? I use the cultivated seaweed. They are not the wild ones. They are cultivated in area, and they are harvested every 30 days, like that. Uh, is my answer clear, or is there any question?
That's all, Ibu Ratih.
Yeah, that's all.
Okay, thank you for the
Is it
answer.
clear or not?
Yeah, very clear. And okay, Pak Murah also like say thanks for the response for your answer.
Thank you.
And I want to recall of Fadli Zainuddin. May you fill in room to answer the question. Pak Fadli. Maybe Pak Fadli kan tahu. Maybe. Okay, so the time is up for the discussion, already 10 minutes, I think. Now, the next to, we still in session 1, but on B, just we have 10 center, so we divide two sessions on one for this session. And for the second one session, side B, we have 4 presenter. Ibu Dia Wahyuni Abida, Ibu Lia Amanda, Ibu Tuti Hakati Siregar, and Bapak Muhammad. And now I would like to invite Ibu Indah Wahyuni Abida. Are you already in room? Okay, so I have information. I just have the information from the IC. Ibu Indah send the video to present the station, and please open the video.
Good morning. In here, I will present the results of the research with topic bioaccumulation of polycyclic aromatic hydrocarbons (PAH) in simping scallops Placuna placenta from the waters of Soca and Unju Bangka, East Java. 1. Introduction. Indonesian waters receive significant pollutant inputs, including heavy metals, PAH compounds, organic and inorganic waste from human activities such as fishing, transportation, ports, industrial operations, waste discharge from land, and human settlements. PAH, polycyclic aromatic hydrocarbons, are a class of hazardous pollutant compounds found in the environment known for their carcinogenic or mutagenic properties in organisms, including humans. The scallop, a non-selective filter feeder, also exhibit a sedentary lifestyle, making it an excellent candidate as a sentinel organism based on its characteristics. Local fishermen in Ujung Bangka and Socha often rely on these scallop shells from coastal waters, highlighting the need for knowledge regarding PAH bioaccumulation in these cells. This knowledge will prove valuable in terms of providing information and facilitating efforts to manage scallop resource effectively. The objective of this study is to assess the bioaccumulation of PAH or polycyclic aromatic hydrocarbons in scallop cells or placuna placenta collected from the waters of Socha and Ujung Bangka in East Java, Indonesia. 2. Material and method. Sampling took place at each station on two separate occasions in December 2018 and January 2019. The research involved collecting samples of water sediment and scallop placuna placenta from two designated stations known as scallop habitats. These stations, namely Station 1 at Ujung Bangka Gresik and Station 2 at Socha Bangkalan, were selected for the study. Water samples were collected using a 100 ml Van Don water sampler, whereas sediment samples weighing approximately 500 grams were obtained using an Ekman grip tool. Scallop samples were manually collected from the Socha station and using a stretching tool at the Ujung Bangka station. A total of 10 scallop individuals measuring 7 until 10 centimeters in size were collected. The analysis of PAH compounds was conducted using the KCMS tool, a method that involves the separation of organic compounds through gas chromatography or GC and the utilization of mass spectrometry or MS to examine the molecular structure of the analyte. This analysis was carried out at the chemistry laboratory of Muhammadiyah University of Malang. The geographic sampling location can be observed in the map depicted in Figure 1. 3. Result and discussion. The high concentration of PAH compounds in Ujung Bangka waters is likely due to presence of numerous fishing boats that generate diesel or fuel waste, as well as the high level of industrial activity in surrounding area. These compounds can be carried away by currents since the coastal area directly faces the Java Sea, and there are also offshore oil mining activities in the vicinity. On the other hand, at the location where placuna placenta scallops were collected in Socha waters, there were only a few fishing boats present, as it is a small boat crossing point to Gresik, resulting in a relatively lower number of PAH type and a smaller total PAH value compared to Ujung Bangka waters. The top 5 PAH components, which include naphthalene, asenaptane, benzogipirylene, 2-methylnaphthalene, and 1-methylnaphthalene. It was found that the water sample from Ujung Bangka contained 18 types of compounds, while the Socha water sample contained 15 types of compounds. Table 2. The top 5 PAH components on sediment sample from Ujung Bangka, which include naphthalene, asenaptane, benzogipirylene, 2-methylnaphthalene, and 1-methylnaphthalene, whereas in Socha different on asenapthalene. Table 3. From the table above, it can be seen that the PAH values contained in the bodies of claims between biota from Socha and Ujung Bangka waters are almost the same, namely the types of naphthalene, 1-methylnaphthalene, 2-methylnaphthalene, and asenaptane. Furthermore, After calculating the level of PO concentration, the highest value was obtained for the types of benzo, urantane, benzoylgibberellin, and the phenanthrene that was able to accumulate was higher than the waters in Ujung Pangkah. While the sediments that were able to accumulate higher were benzoylgibberellin, phenanthrene, and fluorine. Discussion. The accumulation of PAH from water and sediment in clam cells placuna placenta has experienced significant development where anthropogenic activities such as industry, human settlements, and on land have the potential to affect water quality. PAH present in the environment have the potential to be harmful to the life of organisms as indicated by the ratio of ERL, low effect range, and ERM, medium effect range. Values which determine the environmental quality of PAH in sediments based on Hamilton et al. 2015. The lipophilic nature of PAH leads to the accumulation in the lipid tissues of organisms. Once inside the body, particularly in the liver, PAH are metabolized by cytochrome P450, an enzyme assisted by epoxy hydrolase. The metabolism process results in the formation of epoxide derivatives, dihydrodyl derivative, and cation radicals based on Cavalieri et al. 1995. The threshold value established by the Indonesian government based on KMNLH 2004 for the benefit of marine life is 0.003 ppm or 3 ppb. The PAH levels found in water samples from Ujung Pangkah and Socha waters were below the threshold value, with concentrations of 37.08 ppb and 25.91 ppb, respectively. 4. Conclusion. The PAH value found in the bodies of scallops collected from the waters of Socha and Ujung Pangkah were nearly identical, comprising naphthalene, 1-methylnaphthalene, 2-methylnaphthalene, and acenaphthalene. Disparities in the occurrence and concentration of PAH between different sampling stations in water and sediment can be attributed to the hydrodynamics of the respective bodies of water and the amount of PAH contaminants being introduced. The PAH PO concentration value in scallop cells was higher in water when compared to sediment. Thank you.
Okay, thank you for the video to in that what we did that. And now I would like to invite Ibu Lia Amanda. Are you already in the room, Ibu?
Hello.
Okay, so please open the presentation, Lia, and the time is yours, Ibu Lia.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Permit me to introduce myself. My name is Lia Amanda, a student from IPB University. And in this occasion, I will present my study with the topic of you can show the title. I'm sorry. Yes. The topic is food and feeding habits of Tertambra or Valenciennes 1842 in Cibareno River, West Java, Indonesia. Next, please. The background of this research that Cibareno River is river that flow between Banten and West Java province. There are several fish families that occupy in this river. One of the species from this river is Tertambra. Tertambra is a freshwater fish with high economic value in Indonesia and Malaysia because this is a consumable fish that has sweet taste and rich of fish oil. One individual can reach more than 50 kilograms with a length of up to 100 centimeters. So their large size makes them often referred to king of river. And then Caringan weir is a weir construction that raises the water level to irrigate the irrigation area above the base elevation of the Cibareno River. However, the construction of this weir has the potential to negatively impact to the habitat and fish population in the river. Information on fish and food habits can be used to determine the food composition and feeding behavior of fish, also useful to support fisheries management activity such as aquaculture and conservation effort. Next, please. The objective of this study was to examine the food and feeding habits of Tertambra in the Cibareno River as the basis for conservation effort pertaining to the possible fragmentation of the river resulting from the development of a weir. Next. Tertambra sampling was conducted during 4 months, that is May, June, July, and September in 2022, and fish samples were taken in the Cibareno River using the purposive sampling method with 6 sampling stations. The determination of station point is based before slide. Yes. The determination of station point is based on consideration of the construction of the Caringin Dam on the Cibareno River. Station point 1, 2, and 3 are upstream, and station point 4, 5, and 6 are downstream, while Caringin Dam is between station point 3 and 4. Furthermore, Tertambra sample then analyzed at the Macrobiology Laboratory, IPB University. Next. Data collection of water quality of Cibareno River is known by measuring and observing several physical and chemical water quality parameters of Cibareno River. The method and test of measurement of water quality parameters are presented in the table. Next. Data collection of fish was conducted in the morning from 7 to 11 a.m. using gill net, cast net, bamboo trap, and jellyfish trap. And then fish samples that have been obtained are then preserved using 10% formalin. And fish were put into aquariums to remove formalin from the fish. Furthermore, fish morphometric were measured. After that, the fish were dissected and fish food was analyzed using index of preponderance method. Next. The data analysis used is language relationship, condition factor, index of preponderance, breadth, relative brood length, mode size, relative mode size, and relative area size. Next. This part is result and discussion. Water quality condition based on physical and chemical parameters so that all the parameters are in accordance with the water quality standard and are suitable for Tortambara habitat. Next. The figure of distribution of Tortambara based on station show that distribution of the most fish is more in downstream than upstream. This can be caused by food availability in upstream is less than in the downstream. In addition, this condition can also caused by the construction of Charingin Dam between Station 3 and Station 4, which is the construction of dam cause difference in terms of diversity between the upstream and downstream part of the river. Distribution of Tortambara based on time. So the Tortambara was mostly caught in September and the least in July. This is due to the different season in July and September. That September is the rainy season in West Java and July is dry season. It causes main food that insect in the rainy season are more abundant than the dry season. Next. Length distribution of Tortambara show that Tortambara chalk in the Cibareno River was dominated by small size, and previous research by Rahmatika et al. 2002 indicates that the size of Tortambara chalk in Cibareno River was not much different in 2002, and the table graph of parameter of Tortambara so that Tortambara in the Cibareno River has an isometric growth pattern, or B is 3, indicates that Tortambara has balanced weight growth with length growth. Then condition factor of more than 1 indicates that the fish receive a fairly good food intake and have a slightly fat body shape. Next. This is food type identified of Tortambara in Cibareno River. Next. This is index of preponderance that show that main food of Tortambara is water insect, and supplementary food is nematodes and phytoplankton, and additional food is plant seed. Next. This is index of preponderance based on time, so that main food or insect is more frequent in May, June, and September, which is a rainy season. Next. The niche bread in table, so the Tortambara in the Cibareno River has a food niche of 0.6 that indicates there are many types food consumed by the species.
Only 2 minutes left. May you pass your presentation or may you move to the conclusion? Thank you.
Okay. Can I next? The conclusion is the main food of Tortambara in Cibareno River is aquatic insect with nematode and phytoplankton as complementary food and additional food in the form of plant seed. This fish is a bentopelagic fish that feeds during the day or diurnal with many food preference and is not selective in choosing food. Investigation is in situ conservation by constructing of suitable design fishway to facilitate fish migration, restricting on fishing and sand mining in the Cibareno River, and ex situ conservation by conducting hatcheries or farming in pond for restocking. Thank you for your attention. I'm return to moderator.
Okay, thank you very much, Ibu Amanda, for giving us informative and interesting presentation about food and eating habit for the pertama. And now I would like to invite Ibu Tuti Hatati Siregar. Are you already in
Yes,
room?
yes, I'm here. Yeah, I'm here.
So wait a minute for the presentation.
Excuse me, can I share my own screen, please? I added some changes this morning. I don't know if you already received it or not.
Please, Ibu, you can
Okay, thank you. All right, thank you. I would like to present my result about arsenic uptake and metabolism by a marine cyanobacteria. This genetic of the cyanobacteria is already fully sequenced. So it's we call it PCC 7002. I'm from National Research Agency on Innovation from Research Center for Food Technology and Process in Yogyakarta. So to give you a bit of introduction, how arsenic end up in the marine environment. So arsenic can be coming from the natural resources states, volcanic activity, withering, and sort of. Also, arsenic can come from the anthropogenic activity, such as mining, because in the nature, arsenic is never found by itself. It's usually combined with other metal, such as gold, lead, and copper. That's why when people doing mining, usually the side product or the pollution that come up from the mining will be arsenic. The other thing is arsenic also used in several industrial states, alloy or battery, and also used in some of human activities, like in urban activities. For example, we use it in the farming to make pellet. Therefore, all of these source of arsenic is usually going to the riverine system, and then at the end, it will be end up in the ocean. So arsenic in the ocean, it's even though the ocean is very, we call it pristine environment, even though it's very clean, like the algae, either is a microalgae or a macroalgae, so they don't have like metabolic system to differentiate. So usually, I hope that, can you hear me? Okay, so usually arsenic will be taken up by this primary producer from the marine environment, and then they take they couldn't differentiate between nutrient and arsenic, because arsenic is very typically similar to the nutrient, say it's phosphate or sulfonate or anything like similar to the nutrient, or even nitrogen in the marine environment. So we remember that this morning our keynote speaker talking about resilient. This type of organism in the ocean, cyanobacteria or macroalgae or microalgae, they are very resilient to the pollution. In this particular arsenic, they changed, or we call it transform arsenic from the very toxic inorganic arsenic that's coming from the environment, and then they change it into something like mild, we call it detoxification process. And then that's how arsenic then come up to the cycling of food cycle. And then the herbivores like eating arsenic from the primary producer, and then the high trophic level of the animal then eat the herbivores and then go on its cycling in the marine environment. There are so many research related to arsenic. We know that from the arsenate, we call it arsenic 5, and then arsenite, we call it arsenic 3. These two present in marine environment and then taken out by a primary producer, and then changed through the metabolic process, and then change it to the organic arsenic. First, we call it like methylated arsenic, and then at the end change to the sugar form. Even though there are so many research in this area, there are so many gaps that we need to know, and how the uptake of arsenic and then how it changes. We still don't know. We still have a gap we still lack of knowledge in that particular area. To give you a bit of knowledge about what arsenic that I talked today. So this is the arsenic in the environment, and this is the sugar form that transform in the primary producer, and then this is they usually come up from the marine animals. So to investigate the gap in this research, there are two objectives we have in here. To investigate the uptake of inorganic arsenic by cyanobacteria, I use here the anexic culture. So we use batch cultures. And then second, I need to identify the changes of arsenic during the growth phase of the growth phase of cyanobacteria in relation to the arsenic concentration in the media. So to conduct the research, this is the design that I used in this particular research. So I have here control experiment, and so I didn't put any arsenic level in here, and then I have 3 different arsenic level, 5, 10, and 15 microgram per liter. So I call it like small, low, medium, and then high exposure. The lowest exposure is similar or mimic to the what we have in the marine environment. So before I conduct this research, of course, I have pre-culture, so to see how long that the experiment will take. So then I know that how long I could expose the cyanobacteria into arsenic. So we call it like so the best growth level of the cyanobacteria. And then at the end of the experiment, we harvest the cyanobacteria and then dry it, and then using several preparation, and then check it to the instrument, and then we analyze. So this is the method. So for the total arsenic, I only use what we call ICP-MS. And then for the speciation, because arsenic is never found, just like as I told before, arsenic will be changed in the metabolism during.
Your motivation, or maybe you move to the conclusion because your time is set only 2 minutes left.
All right. Okay. So so this the how I prepare the sample. And then from this, from the pre-experiment, I know that the best growth level of the bacteria, the cyanobacteria, is less than 10 days. That's why I then approach the experiment. It's like 1 week. And then this is the result from the uptake of the arsenic. So we can see here the more arsenic introduced into the media, the highest the arsenic level in the culture. So to make sure that the analysis is in line, so analysis is reliable. So I also have a certified reference material using in the analysis. So we can see here after 1 week of the exposure, there is no much left arsenic in the media. And then it's already taken up by all the cyanobacteria. Sorry. And then this is the result of the speciation. So we can see here the highest level of the arsenic, it's already changing, not into the, so it's not anymore in the what you call inorganic arsenic. It's changed already into sugar form. We call it here, it's phosphate sugar, and then this in line.
Your time is over.
Okay. All right. Right. So can I do the conclusion a bit? Can I do the conclusion or just enough? Okay, so to conclude that the cyanobacteria has an important role in arsenic cycling. So it can absorb or it can take arsenic from the environment, and then the cyanobacteria are able to uptake the arsenic up to 90% of arsenic level in the environment. And then the cyanobacteria also can transform arsenic from inorganic arsenic into the sugar arsenic. So I would like to thank Dean Price and Adam Gunn who helped in the preparation of the analysis. Thank you.
Thank you very much to Ibu Reda for giving us an informative and also the interesting presentation. And now I would like to invite Bapak Fauzi. Are you able to hello? Okay, I think Bapak Fauzi is upset on this presentation. Upset on this presentation. So now we can move to discussion session to question and answer. So I would like to have 3 brief questions from the participants who joined with us in Zoom or in class here. The student from Udayana University. May you raise your hand for the participant in Zoom or write down your question in the chat. Okay, we have question from the student in class here. So please mention your name and institution and also whom your question is.
Thank you. Hi. Good afternoon for all participants here. My name is Nicomas Sasaloka. You can call me Udayana. I have question for Mrs. Lia Amanda. My question: how can Institute conservation method for the fish species be carried out? And that's my question. Thank you.
So please tell us about the Institute conservation. What do you think about the Institute conservation for Kamra? I think that's my question from Sai. Ibu Lia, Amanda, are you stay in the room? So the question is, so Ibu Lia didn't hear the question clearly. So the question is, how can the institute conservation for camera? I think like the mechanism or the structure or how can the method or something for institute conservation for camera in your sampling station. Is the question clear, Ibu Lia Amanda?
The question is how to conservation of Tertamra.
Institute conservation in your
I'm sorry, my speaker is not good, so I can't hear clearly the question.
So we will write the question in the room chat, and later you can
Okay.
answer.
Please chat the question. I'm sorry.
Question is up to chat room. Please, we will Amanda to answer the question. Thank you.
Okay, thank you. How can institute conservation method? In the suggestion, I show that institute conservation method is you can construction of a suitable design fish way to facilitate fish migration, and then restricting on fishing and sand mining and Cibareno River, because conservation institute conservation is to protect the Tertamra in that natural habitat. Thank you.
Thank you for your answer, Ibu Amanda. I think you clear about the institute conservation for Tertamra. And now we have Bapak Widianto as head.
Yeah.
Please mention your institution and also to whom your question is for. Thank you.
Yep. I'm going to ask to Miss Lia Amanda. Miss Lia Amanda, my voice is clear.
Yes, I think.
Because I saw in your table you use brightness. I think you have to use water transparency. Is it? Yeah, isn't it? Yeah, water transparency. And then my question is, on your results of feeding by Tortamra fish, yeah, you have addition food. So what are the addition food which has been put by Tortamra, which has your analysis? Thank you.
Okay, Udayana, please respond.
I'm sorry, with the question.
Okay, I will. What are the additional food which has been food by Tortamra fish?
Okay, additional food in of the tambra is plant seed or biji tumbuhan. That is what I mean.
Repeat.
Yes, plant seed.
Bapak Sugianto can hear you clearly, I think, because we
Yes, 50% clear. I think it's enough. And then, yeah, it's okay. Thank you.
okay, thank you, Pak Sugianto and Ibu Yamanda. We have the next presenter. We have the next participant who raise the hand, and please, Bapak Danandaya, to open your mic and please mention your name and also your institution and to whom your question is.
Yeah, thank you for letting me asking my questions. So my name is Gita, and I would like to I'm from Udayana University. I would like to ask the Mrs. Tuti Siregar about the presentation that she just introduced us. First, I would like to thank you for giving me the this information. This is very refreshing, and I know so and in the meantime, there's a lot of algae and bacteria alternative strategy that has been used for environment to tackle environment pollution such as carbon dioxide uptake and oil spill. But my question is for this particular cyanobacteria, so it is said that it will be converted, the arsenic will be converted into arsenic sugar. However, is it still harmful to the environment? Acknowledging that cyanobacteria at the end will decay and degrade into the environment. Thank you.
Okay, Ibu Tuti, silakan.
Yeah, it's clear enough. Thank you. Thank you, Mas Gita. That's a very interesting question. Yes, I actually so this is part of my thesis actually. And then I use this cyanobacteria like to do the pre-experiment for the whole experiment in my thesis, in my PhD thesis. Because I'm not using this data for the main thesis, so it's just too pity to like leave it behind. So why not? I'm using this data for this conference. As for your question, yes, that's right. Cyanobacteria can detoxify inorganic arsenic from the marine environment and then change it to the arsenic sugar. Until now, we still don't know whether this type of arsenic sugar is harmful to the human metabolism or not. So we call it here. If we call about the harmful food harmful or food hazardous, we always concerned about the human, right? So to the environment, yes, at the end, arsenic sugar, it's cyclic. So when the alga decay, so there is other bacteria collaborate with the primary producer, and then change back to the original of the inorganic arsenic. But most of it like will be what we call it will be excrete from the metabolism as it is. So it's not very dangerous, but some of it will be changed back into the inorganic arsenic. So in terms of the ecology perspective, yes, there is still hazardous in there. So that's why in my whole picture of the experiment, I also see how the fate of arsenic from the inorganic and then change to the what we call it sugar arsenic, and then change to the arsenobutane, how it end up back into the environment. Yes, there is still ecology hazardous in there. But to human perspective, if we call it the food hazardous, if the what we call it, if the animals like say mussel, crabs, or fish taken up arsenic from the cyanobacteria or algae, it's not really pose hazard to human for the food safety perspective, but for the ecology, there's still hazardous in there. Is that answering your question?
Yeah, thank you so much, Ibu Tuti.
Yeah.
Okay, I think the question and the answer is clear. And now we still have 3 minutes for the presentation. So we still have a question. Still going. I think that is all question from the participant. And finally, we come to the end of the season 1 on room 4. And I would like to thank the presenter for the fruitful insight and the participation, for the constructive comment and also the question. And please give an applause to the presenter and the report. So big thank you from me, and see you on next session. Now we move to the agenda for lunch and break time, and later we will start session 2 at 1:30 in Lantai. Thank you.
Thank you.
Swastiastu. Good afternoon all, and hopefully you are safe and have a good connection. Dear all honorable, the center and also the participant on this session 2 on room number 4 in the second international seminar on peace and fisheries science. I'm Yoga from Warmadewa University. I will lead this session as the moderator. I am pleased to meet you here and welcome to all of you. Bapak dan Ibu, ladies and gentlemen, before the presentation starts, let me tell you how this presentation goes. First, I will invite each speaker to present for 10 minutes, and after that, followed by the discussion session. For this session, we have 6 presenters. The first is Ibu Dian Pamunasari Anggraini. And second, Bapak Prawira Rajan Bolon. The third is Ibu Cecilia Eka Aisyah Misami. And then Ibu Sekila Nur Faika. Sorry. And the fifth is Ibu Jamulia Said. And the last is Ibu Tuti Kadari. I think all the presenters are already in room, so we can start the session by Ibu Dian for the first presenter. I invite Ibu Dian.
Yes, I'm here, Pak Yoga.
Okay, thank you.
Is
Ibu.
my voice clear?
Yes, your voice
Okay, thank you.
clear. And the PIC will help you to share the screen of your presentation. Okay, Ibu Dian, the screen is yours.
Maybe in slideshow, please. Okay, okay. Thank you, moderator, for the time that is given to me. I'm Dian Pamunasari Anggraini, and this is our team. And now I will present the presentation entitled Growth Parameters of Chana striata in the Musi River, Bengkulu, and South Sumatra. Next, please. Next slide, please. Okay. The Musi River, which is included in the fishing area of the territory of Inland Water Fisheries Management of the Republic of Indonesia, or we call it WPPN RIPD number 438, is a complex river system with a length of more than 750 kilometers, consists of strong currents at the foot of the mountains, lowland, freshwater, and brackish water estuarine parts. The river, the Musi River originates from the Bukit Barisan Mountains in Bengkulu province and ends in Sungsang village in Banyu Asin Regency in South Sumatra province. Next, please. Yes. A river basin from a fishery standpoint has a very vital function.
Recording in progress. Recording stopped.
I apologize for the presenter. We have technical trouble before, so we start the presentation from Udayana. And please the IC to share the screen of the presentation. Udayana, are you
Yes.
right? Okay.
Yes, I'm here.
So we can.
Okay, please, and then slideshow, please. Okay. Maybe directly to the next slide. In the third slide. And then next, please. Okay, thank you. A river basin from a fishery standpoint has a very vital function. Utilization of fisheries is like a coin with two sides. On the one hand, it provides benefits, namely as a source of protein, livelihood, and a source of regional income in the form of water auctions and fishery fees. But on the other side, it also leaves a myriad of problems, such as overfishing and habitat destruction, which in turn can have an impact on decreasing fish production. Due to the demand for striped snakehead or Chana striata as a raw material for making special Palembang food, such as kokrus, or we call it pindang, fish cake, or we call it pempek and crackers, or kerupuk, the exploitation rate becoming high and triggering continuous fishing by the fishermen. But the information related to the stock is not widely known. Next, please. So the research intends to examine the provision of striped snakehead in the Musi River as a basis for taking policies in river management. The research was conducted in March, April, and June to September 2018. And this is a map of the location. There were 6 stations, 2 stations in the upstream, 2 stations in the middle, and 2 stations in the downstream. Fish sampling is carried out by fishermen using fishing gear in the form of nets with various mesh sizes. Will be identified to the species level and also includes calculation of length and total weight of fish. Next, please. Next slide. Okay. These are all the equation that we use for the calculation or data analysis. The first is growth parameters. This is the equation. We can see it in the slide. And then the second is total mortality. The total mortality rate is defined by a catch curve from FISET program. And then the third is natural mortality. Next, please. The fourth is mortality due to capture. This is the equation. F is equals Z minus M. And then the last one is exploitation rate. The exploitation rate is determined by comparing the mortality due to capture to the overall mortality rate. And the equation is E is equal F divided by Z. And this is the condition. The condition, if E is bigger than 0.5, is overfishing. And if E is smaller than 0.5 is underfishing, and if E is equal 0.5 is optimal utilization. And then the result and discussion. Result of growth parameter analysis of the striped snakehead fish using the FISET program. We obtain L asymptote is 53.09 and K is 0.28. This is low category because it's smaller than 0.3, and this is the equation for Batanavilan growth equation that we obtain. And then number 2, from the curve, the value of total mortality rate that we obtain is 0.95 per year. Is smaller than 1. The greater the value, the higher the mortality rate of the fish in that place. And then mortality due to capture, for F, is 0.28. From the equation, F is equal Z minus M. So the value is 0.28. And then the last exploitation rate, from the equation, E is equal F divided by Z. The value is 0.29. And from the category is underfishing. Next slide, please. Okay, the conclusion. The growth parameters of striped snakehead fish or Chana striata with a low growth coefficient, and then the mortality due to capture is lower than the natural mortality rate, and the exploitation rate is lower than the optimum exploitation rate. According to research finding for striped snakehead fish in the Musi River, proper management of this problem necessitates are controlling fishing activities, regulating fishing grounds with due regard for spawning grounds, and prohibiting the capture of small fish in order to produce sustainable fish resources. Okay, that's all my presentation. Thank you for your attention. I give back the time to the moderator. Thank you, Pak Yoga.
Thank you very much, Ibu Dian, for giving us an informative presentation about the growth of Chana striata in Musi River. And now I would like to invite the second presenter, Bapak Prawira Bolon. Are you already involved?
Yes, I'm ready, Pak Yoga.
Okay, so the screen is yours.
Thank you. I'm waiting for my presentation slides. It will be displayed by the committee, right?
Okay, wait a moment.
Okay. Thank you, Mr. Yoga. It's nice meeting you in this seminar. Hi, everyone. I do hope my voice reaches you well. My name is Prawira Tampu Bolon. I'm a researcher in Research Center for Fisheries, National Research and Innovation Agency, or BRIN. For this occasion, I would like to present the report of length-based population parameters of swordfin scat, or Decapterus macrosoma, in Bali Strait waters, Indonesia, that I wrote together with Professor Oh Chul-ung from Pukyong National University, South Korea. Next slide, please. My presentation today is going to be divided into three parts. The first one is the introduction that mentions the background and the objectives of this study. The second one is the material and methods. It will be talking about the research and site period, and then how I collected the data in sampling method and how I analyzed the collected data. And the last one is the result and discussion. We will talk about the landed fish composition, the length structure of the swordfin scat, the length-length relationship, the length-weight relationship, the length at first maturity, and the length at first capture. Next slide, please. So the background of this research is, since Indonesia has a very wide area of waters, the fisheries management area was divided into 11 areas. One of the fishing ground is in Bali Strait, which is in VMA 573. The most active fishing gear in this area is Pursein, and the most captured fish is Sardinella lemuru. For such a long time, the Sardinella lemuru become the backbones of the fishermen in this area. Next slide, please. As we can see from the graph on the left, but since 2016, the Sardinella lemuru tends to be declining. Since the Sardinella lemuru is disappearing, the fishermen depends their financial to the other fish, which one of them is scat or Decapterus. The swordfin scat or Decapterus macrosoma is one of the scats that was caught in this area. The swordfin scat, unfortunately, only little information about this fish based on the population parameter. So the objective of this research is to reveal the length-based population parameter of Decapterus macrosoma, such as length-length relationship, length-weight relationship, the first size of maturity, and the first size of capture fish. Next slide, please.
Iya, nggak apa-apa. Bisa Pak tadi jangan Pak.
Next slide, please.
Sekolah aja dari sama kayak gitu.
The fish samples were collected from Pengambangan Fishing Port. It is in the west part of Bali Island. The samples were collected in 7 months, in June to December 2018. In this slide, we can see the landing situation. The fish were collected in the basket, and this is the percent around the Bali Strait waters. As we can see, the percent is in two-boat percenter. Excuse me, I think I heard something, which is not my voice. Thank you. Next slide, please.
May you unmute your
I still hear some leaking voice. Okay.
Yang ulat ya. Di sini kita ke Poland tadi, lengket langsung.
Should I just continue or we wait for the leaking voice?
Udah gitu aja. Mulutnya. Mulutnya ke atas. Karena itu asli mulutnya.
Moderators, should I continue or we wait for the leaking voice to be off?
Yes, please. I can continue.
Okay, I can continue, right? There were two approaches. Next slide, please.
Excuse me, I think it's disturbing us.
Yeah.
Maybe host can mute this voice.
It's kind of distracting me too.
Yeah.
Can someone ask him to just unmute the voice? Okay, thank you. There were two approaches used to collect the data. The first one is enumeration and the second one is monthly visit. For enumeration, we put an enumerator in the fishing port to record the daily composition of the landed fish and then pick some of the fish randomly to be measured for the foc length. The enumerator is the port officer that we train to be able to identify the fish, especially the Decapterus, which is kind of similar one to the other, and also training for recording the data. For monthly visit, I came every month to the fishing port. At this time, I did more detailed measurement, including the standard length and the foc length, and also dissected the fish to determine the sex and the maturity stage. Next slide, please. We collect these three types of the length measurements: the standard length, the foc length, and the total length. Next slide, please. Since Decapterus macrosoma is not sexually dimorphism and dichromatism, the sex and the gonadal maturity were determined visually based on the color, form, and the size of the gonad. The detailed criteria was given in the table. After that, we classify the fish into two groups. The juvenile is when the fish is still in mature and maturing stage, and we group them as adult fish when it reached maturity, spawning, and spent fish. Next slide, please. For the data analysis, we use regression and logistic model. For length-length relationship, we use linear regression. For length-weight relationship, we use power regression, and we use logistic models for the length at first maturity and the length at first capture. Next slide, please. During the study, we found there were three kinds of scuds landed in Pengambangan Fishing Port. They were Decapterus macrosoma, in local name we name it as layang deles, Decapterus russelli or layang banyar, and Decapterus choroides or layang anggur. As we can see in the graph, Decapterus macrosoma is the most abundant among the other Decapterus. Next slide, please. From the enumeration, we measured 2,341 fish in 7 months. The smallest fish was 110 mm foc length, and the largest was 220 mm foc length. Next slide, please. And then we randomly took 420 fish to be measured more detailed and dissected. From this graph, we could see that the length characteristics of every length measurement, the average for the standard length was 146.9. The foc length was 153.69, and the total length was 165.82. Next slide, please. The total length, foc length, and standard length relationship is close to each other. We could see from the equation that the R-square for each relationship is above 0.98. Almost reach 1 for every relationship. Next slide, please. From the length-weight relationship, we also could see that all type of the length measurement also have a close relationship with the weight. The total length, poke length, and the standard length are good tools to estimate the weight. Next slide, please. We analyze the length-weight relationship further by separated the analysis into the sex and the maturity stage. It is still shown that the R square is more than 0.9 for every regression. After testing the B slope with t-test, we found that the growth pattern of the scat is hyperallometric for every sex and maturity. The other researchers also reported that this phase tends to be hyperallometric. Next slide, please. By regarding the 50% of the proportion for the mature in the logistic model as the length at first maturity, we found that the length at first maturity for the male was 164 millimeters, while for female was 162 millimeters. Next slide, please. The length at first capture was 184 millimeter. This is bigger than the first size of maturity. These scat were harvested by the person after they grow and reach the maturity. That means they got a chance to spawn at least once in their lifetime, which is good for the management. Next slide, please. As the conclusion, we know that all type of the length measurement relatedly strong to the body weight. The growth pattern was hyperallometric. The length at first maturity was 164 millimeter for length and 162 millimeter for length for females. And the length at first capture was 184 millimeter for length. Next slide, please. I would acknowledge that the fund for the data collection is supported by the Research Fund in Research Institute for Tuna Fisheries in Ministry of Marine Affairs and Fisheries Indonesia. Next slide, please. That's all that I can bring for today. Thank you very much for the attention, and discussion is welcome. I bring the stake back to the moderator. Thank you.
Okay, thank you so much, Bapak Prawira, for giving us the interesting information and also the informative information to us about motorization for the recovery of customer in maritime water. Now, I would like to invite the third presenter, Ibu Cecilia Eka Aisyah Isami. For Ibu Cecilia, are you in the room already?
Yeah, I'm in the room already. Hello?
Okay, hello. Yeah, we can hear you.
This,
Okay, you please.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. My name is Cecilia Kaishari Sami from IPB University, and today I will present my research about reproductive aspect of the Japanese sapling bream, Hemipterus japonicus, foot 1791 in the southern Java waters, FMA 573. Next slide, please. Okay, the background of this research is FMA 573 covering the water from southern Java to southern Nusa Tenggara, which directly border the Indian Ocean. The water of southern Java generally have abundant catches with a considerable number of demersal and pelagic fish being caught. Hemipterus japonicus, also known as a Japanese sapling bream, is a demersal fish belonging to the Hemipteridae family and widely distributed in the Pacific and Western Indian Ocean. Japanese sapling bream live at depth ranging 5 to 80 meters. Octavini et al. 2016 said the high market demand has made Japanese sapling bream one of the target catches for fishermen in the Bay of Banten. To date, there have no studies on Japanese sapling bream in water of southern Java, making this research highly valuable in understanding the condition of Japanese sapling Java. Next slide, please. Research objective. The aim of this study was to determine of the reproductive characteristic of Japanese satin bream in southern water of Java. The advantage this research provides fundamental data for sustainable management and utilization of the satin bream fisheries researches in the southern water of Java. Next slide, please. The study was conducted from July to December 2022 at the 3 different fishing port research location. There is PPI Binuangan, PPN Pelabuhan Ratu, and PPI Ciwaru. And the laboratory analysis were performed at the Microbiology 2 and the Fisheries Biology Laboratory at PB University. Next slide. Yes. The procedure for this analysis involved sampling 50 fish at each research location every month. The procedure first is the measurement of length and weight of Japanese satin bream. Next, dissection of this fish from the anus for posterior operculum. And the third, determination of sex and visual examination. The fourth is fecundity calculation was performed on female fish at stage 4 using the gravimetric method. And the last one is measurement of egg diameter was conducted to female fish at stage 4 and 3, observed under the microscope at 5 times 10 magnification. Next slide, please. Okay, next is data analysis. The data analysis includes the following sex ratio, calculated by dividing the number of male fish and by number of female fish, and the length maturity calculated using King 2007. Next slide, please. Okay, the next data analysis is gonadal maturity index, fecundity, and egg diameter. Next slide, please. The result and discussion is in the sex ratio. Determining the sex ratio is important to assess to the condition of this fish population in the particularly water area to determine its suitability. The sex ratio of male to female Japanese satin bream was 1 to 1.10. While in the stage 4 or mature stage, the ratio was 1 to 0.78. It's observed that the sex ratio was balanced. States that the difference in the number and size of fish within a population can be caused by several factors, namely difference in reproductive behavior, environmental condition, and fishing activity, as well as the presence of different growth patterns. Next slide, please. Gonad maturation stage. The gonad maturation stage represents the level of gonad development before and after spawning. It is observed that the dominant stage at the stage 1 and stage 2. The number of fish with mature gonads was lower compared to those with immature gonads. According, a good percentage of mature gonads should be above 90%. This research indicates that Japanese satin bream do not meet the criteria for sustainable fisheries research. With such condition, it is a concern that Japanese satin bream may affect the sustainability of the stock in its water. Next slide, please. The length at first maturity. The length at first maturity for the male fish was found to be 146 millimeter, and for female it was 141 millimeter. According to several studies, it has been found that maximum size of male fish is larger compared to female. The size at first maturity varies within a species species depending on genetic factor and environmental factors influenced by food availability, physical density, and water temperature. Next slide, please. Gonadal Maturity Index. GSI is used to measure the level gonadal maturity by analyzing histological database on the weight of the fish gonad. Instead, the increasing GSI value indicates the spawning ground and spawning season. The highest GSI value were observed in October and November, defining the peak spawning season for Japanese headfin bream in the water of southern Java. Next slide, please. Fecundity refers to the number of eggs produced by a female fish. The fecundity values in the study range from 1,110 to 82,310 eggs. Fish with higher reproductive potential have an average fecundity value ranging from 1,000 to 100,000 eggs. Other factors influencing fecundity include fish age, body in size, and then egg diameter. Next slide, please. Okay, the last one is egg diameter. The distribution of egg diameter in female Japanese headfin bream at stage 4 during the study show range between 0.10 and 0.07 millimeter. This graph mature Japanese headfin bream eggs are colorless, spherical, buoyant, and with a diameter of 0.71 until 0.79 millimeter. The research indicated full maturation and readiness for spawning. The frequency distribution of egg diameter indicated the Japanese headfin bream exhibit in total spawner because it has spawning type with a single peak. Total spawner will reach their entire reserve of oocyte in a short period all at once. Next slide, please. The conclusion of this research: the sex ratio of Japanese headfin bream is balanced. The male Japanese headfin bream mature faster than the female. The peak spawning occur in October and November. The reproductive potential range 11,110 to 82,310. And the spawning pattern of Japanese headfin bream is total spawner. So next slide, please. So thank you for. Ya, wassalamualaikum warahmatullahi wabarakatuh. Back to the moderator.
Okay, thank you so much to Ibu Cecilia for your presentation. Yeah, giving us the informative and also interesting presentation about the production aspect of Japanese headfin breams. Okay, now I would like to invite the fourth presenter, Ibu Septenana Maika. Are you already know?
Hello. Is my voice audible?
Yeah, we hear your voice clearly. And you can start your presentation. The screen is yours.
Good afternoon and best wishes to all of us. Honorable to all seminar participants. On this special occasion, I am September Faika from Department of Aquatic Resource Management, IPB University. Allow me to present the result of my research on reproductive biology of the Indian mackerel, Sarigar Kanagurta cuvier, 1817 in the southern waters of Java, FMA 573. Next slide, please. Indian mackerel is a small pelagic fish of the family Scrombidae that lives schooling in the marine waters. Indian mackerel is widely distributed in the Indonesian waters, including the southern waters of Java. Indian mackerel is one of the fish targeted by fishermen. In the southern waters of Java, they have a high economic value, which is marketed fresh or processed into the South Asia. The high economic value of Indian mackerel has caused the fishermen to catch the fish without considering the sustainability of the fish. So, to maintain the Indian mackerel stock in the waters, management of fishing activities can be formulated. Studies on fish reproductive characteristics are important and become a basic recruitment from strategic planning in sustainable fisheries. Next slide, please. So this research aims to analyze the reproductive characteristics of Indian mackerel as a basic information for the management of small pelagic fish in the southern waters of Java. This research provides information about the reproductive characteristics of Indian mackerel, which can be referenced for the managing small pelagic fish resource appropriately and sustainability. Next slide, please. The research was conducted from July to December 2022 at Binuangan Fish Landing Base, Palabuhan Ratu Fishing Port, and Ciwaru Fish Landing Base. Laboratory analysis was accomplished at Biology Macro 2 Laboratory and Fisheries Biology Laboratory, Department of Aquatic Resource Management, IPB University. Next slide, please. The first step in collecting data on the reproductive aspect of fish is measurement the length and weight of fish. Second, fish are dissected to determine the gonad maturity stage. Then the analysis of fish fertility. After that, measurement of egg diameter using a microscope. For the management aspect, primary data on reproductive aspect are used as attribute or indicator. Secondary data relate to fish reproduction aspect from previous research and the regulation. Next slide, please. Data analysis used in this study include line frequency distribution, sex ratio. Next slide, please. Gonad maturity stage. Next slide. Gonadosomatic index or GSI, size at first maturity, fecundity. Next slide. And fisheries management analysis was made using a tactical decision approach on the matrix referring to GAFARIS 2009 and NYG 2014. Next slide, please. The result and discussion, the total number of fish obtained was 138 with 582 males and 456 females. Based on the figure, it can be seen that the highest line frequency distribution on the class interval 190 until 202 millimeter, and the lowest in the class interval 268 until 280 millimeter. Difference in fish size mainly depends on age and environmental factors such as quality and availability of food. Next slide, please. The sex ratio of Indian mackerel population was determined based on the overall number of male and female fish. Based on the chi-square test, the ratio between male and female fish was 1:0.78. The sex ratio of Indian mackerel that are ready to spawn or immature stage 4 is presented in the picture. Chi-square test results show that the ratio between male and female fish is 1:0.79. This ratio indicates that the sex ratio of Indian mackerel in the southern waters of Java is not balanced with dominance of male fish. Unbalanced sex ratio can be caused by fish reproductive behavior, environmental condition, and fishing activity. Next slide, please. The level gonadal maturity of Indian mackerel has 5 stage development, which are stage 1, immature, stage 2, maturing, stage 3, mature gonad, stage 4, late development, and stage 5, span or spawning. The pictures show the percentage of Indian mackerel gonad maturity level in the southern waters of Java. Based on the observation, the level of maturity Indian mackerel gonad in July, August, and September dominate with stage 4. Stage 1 dominate in October, November, and December. Slide, please. The mean GSI of male range from 0.19 to 0.95 and female fish range from 0.45 to 3.41. The highest GSI value of male and female fish were in August. The increase in the GSI value indicates the spawning season. Next slide, please. Based on the analysis, the fecundity value of Indian mackerel range from 27,027 until 87,837 eggs. Based on the results, there is a significant relationship between fecundity and the length and body weight of the fish. This indicates that as length and weight of the fish increase, fecundity will increase. In general, fecundity will increase during growth. Larger fish will have more eggs than smaller fish. However, there will be a decrease in fecundity as the fish gets older. Next slide, please. These are egg diameter distribution, which range from 0.025 until 0.0840. Based on the picture, it can be seen that the distribution of egg diameter has one peak at each level of gonad maturity. So it can be seen that Indian mackerel has a total spawning pattern. Total spawner are those species in which after maturation of the gonad begins, all the egg or sperm which are going to be spawned by the individual fish in a single breeding or period develop synchronously. Next slide, please. The size of the first mature gonad of male fish is 198.5 millimeter, while the size of the first mature gonad of female is 197 millimeter. Based on the analysis, female mature faster than male. Next slide, please. Tactical decision making is based on the reference point of each attribute or indicator. The attributes that were analyzed are fish strength of the proportion of juvenile. Attributes fish size threat show the status size trend in relative fish. The objective strategy that can be used in regulation of exploitation rate or spawning biomass. So the tactic that can implemented is restriction of fishing activities in July, August, and September. Attribute the proportion of juvenile that are captured. Show the status allowed, which means above 60%. So the tactics that can implemented is setting mesh size above 1.5 inch. Next slide, please. So the conclusion is the sex ratio of Indian mackerel in the southern waters of Java is dominated by males. The reproductive potential of Indian mackerel range from 27,027 until 87,837 eggs, with a total spawning type. The female Indian mackerel mature faster than the males. The peak spawning season of Indian mackerel in the southern waters of Java occurs in August. Tactical step for managing Indian mackerel by limiting fishing activities during the spawning season and setting mesh size more than 1.5 inch. Next slide, please. Thank you so much for your attention. I return to the moderator.
Okay, thank you so much to Ibu for giving us the interesting presentation about reproductive biology of target Hanabuta. And now I would like to invite the fifth presenter is Ibu Korea Sahid. So for Ibu Korea, are you already in the room right now?
Oh yeah, thank you.
Okay, wait a moment for the your presentation. Okay, the screen is yours, Ibu Korea.
Oh yeah. Thank you for your for moderator. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name's Jamhuria Said with 5 partners. We are from BRIN, Tirtayasa University, and Halu Oleo University of Kendari. We would like to present our research about the reproductive pattern of Origia sorae, Parenti hadiati 2010, the endemic fish of Muna Island, Southeast Sulawesi, Indonesia, in rearing with varied sex ratio. We see here the performance of the Orygias floweri. Next, please. Introduction. Next, please. Sulawesi Island, according to Hadiati, Sulawesi is a source of endemic ichthyofauna and habitat of various endemic ichthyofauna of Indonesia that are not found anywhere else in the world. One of them is the inland water of Muna Island, southeast Sulawesi of Indonesia. Next, please. We see here several types of inland water of Muna Island that are the habitats of Orygias floweri. Orygias floweri have the common name daisy blue rice fish. The fish is have attractive color and several parts of the body was a bright red and bright blue on the side of the body. So it's have economical value as ornamental fish. The fish is endemic and the smallest Orygias of Sulawesi. Then the fish finding by Indonesian Institute of Sciences on 2010. Next, please. So what's the problem of Orygias floweri? The fish as endangered species according to IUCN 2019, and the biological information is very rare, high exploitation, high habitat degradation, invasive species, and allochthonous activities around the habitat. Also, how to anticipate it, we have to improve the biological information, and it takes effort to conserve it by ex situ or domestication or in situ conservation. So for ex situ or domestication, we can taking fish from the habitat and then cultivating it at ex situ habitat. Next, please. In this study, you want to support a domestication program, how the reproductive pattern, and to study the effectiveness of sex ratio on reproduction in limited number of fish bloodstock. And we would like to obtain the growth pattern from large to adults and the age when the sex differentiation was identified by phenotypic. Next, please. Material and method. Orygias floweri for this research, the broodstock obtained from cultivation from Serang, Banten, 5 months old. And the research was conducted on April 2022 for 3 weeks with 2 replications at Research Center for Limnology and Water Resources BRIN Cibinong Laboratory. We try the sex ratio male to female 1:1, 2:3, and 2:5. Observation component for reproductive viability, such as frequency of spawning, frequency of hatching, number of egg, fertilization rate, hatching rate, number of larvae, egg incubation period. The length size of larvae at certain eggs until adult size. And the eggs of the fish at the time of sex differentiation identified, and the size of male or female. We use an aquarium to rearing the broodstock, and to collect the egg, we put a substrate, artificial substrate that made from tali raffia to the rearing aquarium of the broodstock. And daily we check the telaria that are any egg or not. If there are any egg, we remove the telaria from the rearing of broodstock to the other hatching aquarium. The research we conducted for 3 weeks. Every day, daily we check the same material. And how to measure the body size? We use a transparent for length of body. We use a transparent container and put on the scale such like a millimeter block. And to measure the weight, we use a house balance. Next, please. Table 1 is the reproductive parameter that I said, spawning frequency, hatching frequency, hatching to spawning percentage, total eggs ovulated, fertilization rate, number of larvae, hatching rate, survival rate until 7 days, and egg incubation time. The data that we saw here, it's average of all of the treatment for each sex ratio. Next, please. Result. We see here in table 2 the reproductive ability of Orygia swawarai on first sex ratio. We see here the left one is the reproductive parameter, and sex ratio male to female, A treatment 1:1, B 2:3, and C 2:5. We see here the higher spawning was on C treatment. There are 5 times, and for A treatment there are 4 times average of spawning. We see here to take 5 times spawning, we use 5 female broodstock. And for a treatment, to use 4 spawning, we only use 1 female. And then total egg of this research is the higher from treatment C. But to take 5.6 eggs every spawning, we use 5 broodstock female. But for A treatment, A or we can take 4 eggs from 1 female. But we see here the all of spawning has or hatching. So hatching to spawning percentage all of 100%. We see the fertilization rate. A and B treatment was 100%, and we see here the hatching rate of A treatment of 91.8. So from this data, it's recommended to use the sex ratio male to female 1:1. Okay, please. Next, please.
I apologize, Ibu. Maybe you can move to conclusion because the time less than 1 minute for your presentation. It's okay?
Oh.
It's already 9 minutes.
Oh, yeah.
Just 10 minutes. Okay, please, the PIC move to
Okay, to
conclusion.
Conclusion. How about conclusion? The endemic fish Orygia swawarai for reproduction is recommended to use sex ratio 1:1, and eggs of 5 months was able to reproduce and the larvae able to grow at situ habitat. Sex differentiation begin at 14 weeks, male size tend to be bigger than the female, and it's hoped that it could be developed to support domestication program in the future. So the further research of this is needed. Thank you.
Okay, thank you very much to Ibu Jamria Said to giving us informative and also interesting presentation about the reproductive pattern of Ornithias wari. So now I would like to invite the last presenter, Ibu Tuti Kadarini. Are you in room already, Ibu Tuti?
Yeah. Thank you so much.
Okay.
Yeah. Good afternoon, everyone. Thank you so much. And could you hear my voice clearly?
Yeah. We are able to hear your voice.
Yeah. I'm Amida Suryati with Tuti and another. And now I'd like to present the result of our research about reproductive ability of Lake Ajamaru endemic species Melatonia ajamaruensis, Ellen and Cross, 1980, through the pH increment by adding the different amounts of green muscle cell penaviridis. Could you please share?
Yeah, wait a moment, please.
Okay, thank you. Thank you.
You can carry on.
Okay, okay, good. Okay, thank you very much. Once again, our research about reproductive ability of Lake Ajamaru endemic species Melatonia, Melanotanea, Ajamaruensis, Ellen and Cross, 1980, through the pH increment by adding the different amounts of green muscle cells penaviridis. Next. Rainbow Ajamaru is an endemic fish from Lake Ajamaru, which are located in Papua. Rainbow fish species are endangered in Lake Ajamaru. Their habitat has been damaged due to logging activities, soil erosion, and also invasive fish and land constriction, so that this influence became one of the reasons for the existence of the rainbow fish that was declared extinct and found again. Because of market demand tends to increase very well for Ajamaru fish, so it is necessary to develop it by cultivating it properly and sustainably. The original habitat of rainbow fish is waters that have a pH that tends to be alkaline, such as Lake Ajamaru, pH 6.8 to 8.5. Shell immerse in water will release calcium and react. Next, please. Yeah, that's why. Okay, thank you. The habitat of rainbow in situ is have a pH 6.5 to 8.5. Shell immerse in water will release calcium and react or form calcium bicarbonate compounds, which function as a buffer, pH buffer, or can control pH fluctuations. Besides that, the function of calcium, that calcium is an important element in the development and growth of bones in fish and crustaceans. In condition of low water, water hardness, carbon dioxide becomes scarce. Let's see in my slide, low pH will disturb the ion balance of aquatic organisms, fish plankton, fish osmotic regulation. Next. The Ministry of Marine Affairs and Fisheries, KKP, project a production of 137,000 tons of shells. So they have a project of production, yeah, for mussels. There are about this one, the pity. That means that 1,500 species. Kadarini and Juria, to 2013, the addition of another granulosa shell on Kurumoi rainbow spawning had a higher number. In previous studies to increase the pH value, Kadarini and Juria has a result. The addition 100 gram per 40 liter grams increase to pH 7. Yeah, spawning had a high number of larvae, 355 0.6 in pH 7.3 to 7.5 compared to control 201 plus and minus 182.6 in pH 5.5 to 5.9. And also Udayana and Mustafa et al. That growth and survival of rainbow Melanotaenia parva fish with the addition of green mussel shells. In the spawning of rainbow kurumoi, Kurumoi is a Melanotaenia parva. They have research that effect of growth on rainbow kurumoi with green mussel shells. Next. Oh, sorry. Back. Research purposes. The objective of this study was to determine the best amount of green mussel shells, Panaphyrides, for spawning productivity, and to find out its effect of water quality in spawning media for Adjamaru rainbow fish, Melanotaenia adjamaruensis. Thank you. Next. Materials and methods, time and location. Time in 3 months, and the location in Jakarta Technical University of Fisheries Laboratory, Pasar Minggu, Jakarta. And then we have a container. The container used for spawning was an aquarium measuring 60 by 50 by 40 centimeters with 15 units and 60 units of plastic container were used for egg incubation. Next. Experimental design, completely randomized design, 5 treatments using green mussel in different amounts as follows: A, without any shells; B, 2 grams per liter; C, 4 grams per liter; D, 6 grams per liter each; shells 8 grams per liter each. Treatment repeated 3 times, put in bags, then put into the water media until submerged. Test fish, 30 broods of rainbow fish, 15 females, total length of 6.7 to 7.2 centimeter, and 15 males, total length of 8.2 to 9.3 centimeter. Parent stock to tails per container fed blood worms as much as 5% of the weight. Spawning. Fish were spawned naturally every 2 for 30 days, given substrate fed blood worms and as much as 5% of the weight. parameters and analysis. Spawning frequency, fertilization rates, hatching time, water quality, pH, alkalinity, temperature, oxygen were analyzed descriptively. And also we have analysis by with ANOVA. Next.
I apologize, Ibu Tuti. May you faster your of your time only less than 2 minutes for the presentation.
Okay. So we are So I'm going to conclusion.
Yeah. You can
Okay. Thank you. Okay. So next, please. A conclusion. Okay. Yeah. Okay. Conclusion, the endemic species of Lake Ajamaru, Papua, Indonesia, Melanotaenia ajamaruensis, has the ability to reproduce from the treatment of 2 grams per liter of green muscle cells. Panaviridis obtained the highest fecundity is 537.67, 9.37, with fertilization rate 90.90% and hatching rate 93.33%. The result of the water quality measurement in treatment B showed the range of pH were 7.4 to 7.8. Thank you so much. Time is yours, moderator.
Okay, thank you so much to you, Tuti, for your informative and very colorful presentation because it's a rainbow is your that's about reproductive ability about rainbows. And now we are going to the discussion session by Q&A, question and answer session, and I would like to have 3 brief questions from the participants who already joined in room number 4 or the participants who already joined on our class here in Denpasar. So for the participant who want to ask, please please raise your hand. The participant on Zoom.
Hello, moderator. Someone want to ask you. Someone asked raise their hand. Ibu Tri Ernawati, maybe Ibu Erna want to ask something. Hello, moderator. Could you hear my voice?
Thank you, Pak Charles.
Okay, Miss. Go ahead.
On the computer. Wait.
Can you voice Ibu Tri?
Yes. Is my voice clear?
Okay, I try to hand it over from the. So please, yeah, you can deliver your question to the presenter. Please, Ibu Tri Ernawati.
Okay, thank you, Pak Charles, and thank you for this opportunity. Actually, I want to give a comment for Ibu Dian. In conclusion, the inferred statement conflict between the findings and the objective of the management's options. It said that the F lower than M, and also exploitation rate is also smaller than exploitation rate in the level of optimal. So I think the overall situation for your finding is underfishing or underexploited. So my opinion regarding to management options needs to be encouraged or maintained or maybe increase utilization to the optimal level. Because in your conclusion, it said that, for example, prohibiting for the small fish, and also I forgot. Oh, and also not caught in the fishing in the spawning ground. But in your finding, I didn't see the indication about the recruitment. Indication about the recruitment or growth of the fishing. So maybe you can give me the clarification or a comment for my confusion here. Thank you.
Okay, moderator.
Maaf, Pak.
Yeah, okay. Yeah, go ahead, moderator. Thank you.
Thank you for the question, Ibu Tri. I just joined back to the room, and I have to know to whom your question is going, Ibu Tri. Hello, Ibu Tri.
Yeah, Budian, for Budian.
Directly, I respond, Pak Yoga.
Okay.
Yeah, Pak Yoga, your voice is not clear. Maybe you can use your microphone or something as we can hear your voice clearly.
Okay. Thank you very much, Bu Erna. Maybe I didn't write complete description in my presentation. Maybe it is included in my full paper, but thank you for your suggestion. I will correct my it next. Thank you.
No worries.
Okay, thank you for the response from Ibu Dian. So we still open the opportunity to question and answer question. We'll have time for the discussion session until 3 o'clock in Bali time. So around 5 minutes. Or maybe the participant of the class, then you have a question. Mention your name and your function, and also who your question is.
My name is Jane. I'm from Universitas Udayana. I have a question for Budian. My question is, what size of Tanah Seriata are often shown in Musi River? Thank you.
So the question to Ibu Yan, right? So the question is, the size is often gets in Musi River for Udayana, right? Please to Ibu Yan, you ask your response.
You mean the length of the fish or what? I don't hear clearly.
The size of the gets in Musi River for your patient, yeah, Udayana. The size of the Halo Ibu Yan. Halo Ibu.
The mid length, yeah, the mid length, if I'm not wrong, or hello, hello. The mid length is 25.5 centimeter with interval 25 up to 26 centimeter.
25.5 centimeter to
25 and to 26 centimeter. In middle 25.5 centimeter.
Okay. Thank you, Ibu. Yeah. So the answer is 25.5 to 27 centimeter. The size of the Udayana. Okay, and we still open the activity. Participant who wants to ask the presenter about their tradition. We have one participant, I think, from this class. So, okay, thank you for the time. Okay, thank you for the time. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Before that, I would like to introduce myself. My complete name is Gunawan. I come from Udayana University, exactly Faculty of Marine and Fisheries. I would like to ask to